Good afternoon, and thank you for joining us to the Introduction to Biotechnology webinar. My name is Michelle Parsons, and I'm the manager of the biotechnology portfolio at LASEC. LASEC has been the official channel partner for the Life Science Division at Bio-Rad since 2017. And we ask that you feel free to ask any questions during the webinar, and we'll make sure to answer them at the end of our session. It's with my great pleasure to introduce our speaker for today, Kris Janse, who is the Proteomics Field Application Specialist from Bio-Rad. Thank you very much for your attendance, and over to you, Kris.
Good afternoon, everyone, and thank you for joining us today. So we're going to give an introduction to Bio-Plex Technology. And my name is Kris Janse, and I'm a field application specialist in proteomics. And so what we'll look at today is who is Bio-Rad, Bio-Plex versus ELISA, Bio-Plex Manager software and assays, some practical training, and then some good experimental practice. That's not right. Sorry about that. So Bio-Rad Laboratories was founded in 1952 in Berkeley, California. It's had 60 plus years of strong performance, over 1 million customers worldwide, over 8,000 products. It is a family-controlled business. And the mission of the company is to provide useful, high-quality products and services that advance scientific discovery and improve healthcare. And the president and CEO is Norman Schwartz. So Bio-Rad has products for Western blotting, protein purification, biomarkers, and immunoassays, which we'll be discussing today. Cell biology, PCR amplification, droplet digital PCR, electrophoresis, and general lab equipment. So I'll start on the proteomics side. So the protein expression, multi-analyte detection, that's the Bio-Plex 3D system that's shown in the small image. Then we have protein quantification, Western blotting, and stain-free Western blotting. Protein purification, chromatography, cellular imaging, cell and sorting and analysis, digital biology, droplet digital PCR, food safety testing, which is not part of our division, that's a separate division, genotyping and gene expression, and gene transfer and modulation. So if we look at a classical ELISA, enzyme-linked immunosorbent assay, a Bio-Plex assay works similarly, but it is not an ELISA. So in an ELISA plate, you have the capture antibodies, and then you will have your target protein binding to the capture antibody. You would then have a detection antibody binding to a different analyte or different epitope on the analyte, and then biotin and streptavidin HRP to produce a color change. So biomarker screening is sometimes like working in a black box. Traditional techniques like ELISA can shine a spotlight on one protein at a time. But Bio-Plex illuminates the entire biomarker network, uncovers a broader, more complex picture with increased speed, sensitivity, and dynamic range with less sample. So you can save a lot of time, reduce amount of pressure sample, reduce assay reagent volume, expense, and labor, dramatically increase the amount of information from a single sample, and better correlation of data between different targets as sample analysis is performed at the same moment in time. So if we had a look with the Bio-Plex system to analyze 48 cytokines, you would have 196-well plate. It would take approximately 3 to 4 hours to complete. You would use 12.5 microliters serum or plasma and 50 microliters cell culture supernatant. If you were going to do that with an ELISA-based method, you would need 48 96-well plates. It would take over 106 hours to complete all those plates. You would need 1 ml of serum or plasma and 1 ml of cell culture supernatant. So I'm just going to play a little video quickly. So the beads are coated in infrared and red dye. So that's how the instrument can tell the difference between the beads. So you have antibodies covalently bound to the beads. You have your secondary antibody binding, and then phycoerythrin to generate the signal. And then the beads will follow in sheath fluid. So you will see that the red laser will excite the beads. So the instrument knows which analyte is on the beads, and then the green laser will quantitate the signal coming from those beads. So the beads used are magnetic and uniform in size. The assays use 6.5 micrometer magnetic beads instead of an ELISA plate. And utilizing beads enables detection of multiple analytes in a single well. So each bead region is coated with 2 specific dyes at different ratios. Both dyes are excited at 635 nanometers, but the emission signal differs. So red, 660 nanometers, and infrared dye, 730 nanometers. So thus, the emission signal from each bead is specific and identifiable. So Bio-Plex XMAP technology is multiplex analysis in a single sample, one single working dilution. Less sample is needed, microplate compatible, save time and money, and robust results. Over 35,000 publications are currently available for multiplex XMAP technology. So you can use cytokines, chemokines, and growth factors, immunology panels, inflammation factors, cancer markers, diabetes, cell signaling, isotyping, allergies test autoimmune disease, infective diseases, new COVID kits. So we have NICD using SARS-CoV-2 kits – and homemade assays. So now we'll have a look at Bio-Plex Manager software and assays. So there you see the Bio-Plex 200 instrument with the high throughput fluidics platform and the PC. Then you have the microplate reader, magnetic pro wash station, or you have the handheld magnetic handheld device. But I've seen in the lab people using the handheld device and then they're shaking the plate to get the wash solutions out, and then they drop it on the floor, and that's the end of your assay. And then we have the Bio-Plex Manager software, which is very easy to use, and then the kits. So Bio-Rad, Bio-Plex provides the software, service and support, calibration and validation kits, hardware, the wash station, and they used to provide magnetic beads, but now the magnetic beads need to be purchased from Luminex directly. So you do get quantitative results because you have standards running duplicate, and all your samples are running duplicate as well. So the standards, you can then analyze your results and get quantitative results from the assay. So you have your microbeads in suspension. The first specific antibody is covalently bound to the microbead. Then you can measure your interleukin-2, for example. So the biological molecule is selectively recognized. Then you have second specific antibody binding to a second epitope on the analyte, and that is fluorescent tagged phycoerythrin. So you use the red laser to identify the beads and the green laser to identify the signal coming off those beads. So you have identification, where the red laser is used to identify which bead is being analyzed at the moment. And then you have our quantification, which is targeting the MFI signal coming off the beads. So here you can see interleukin-2, interleukin-4, and interleukin-8, and you see they have different amounts of infrared dye and red dye. So the first one, interleukin-2, is very red. The interleukin-4 is more pinky in color, and the interleukin-8 is darker pink. So you have up to 100 analytes, beads, and we currently have a 48-plex kit at the moment. So the green laser is used to interrogate the label, and the red laser is used to excite the fluorophores at 635 nanometers, and then you will get infrared and the red dyes activated at different wavelengths. And sheath fluid is being used to drive the beads through the system. So the first step is to mix beads with standard samples and controls. The incubation takes 30 minutes. Then you're going to wash the steps between incubations. The first wash step is 2 washes, and all the rest are 3 washes. You add your biotinylated antibodies, incubation for 30 minutes. Then you add your streptavidin and phycoerythrin, incubation time 10 minutes. And then you read in the instrument. So with Bio-Rad assays, it normally takes 4 hours to results with our cytokine assays. And then an orbital plate shake is required for incubation steps, which is not provided, but I believe LASEC do sell these, so you can get these orbital shakers required because you need to shake them at 850 RPMs plus or minus 50. So if you go onto the Bio-Rad website and have a look at Bio-Plex multiplex immunoassay systems, there will be specific information on the Bio-Plex assays, instruments, and software, Bio-Plex instruments, Bio-Plex software, and Bio-Plex standard lot data. And Bio-Plex assay sample prep success guide, hardware and software success guides, and Bio-Plex assay manuals, guides, and protocol library. So we have acute phase proteins, apolipoproteins, apoptosis pathways, diabetes markers, kits for human, mouse, rat, SARS-CoV-2 serology assays, which NICD is using, and cell signaling assays. So it's not only cytokines, but there are a number of other kits that are also sold to customers. And here we have the 48-plex screening panel, but we also have an 8-plex panel, a 17-plex panel, 20-plex panel, 27-plex panel, and TH1 and TH2 panel. And these are the analytes that are included in the 48-plex screening panel. So you can choose whatever format you want to choose. So we had a customer looking at a 48-plex screening panel, but they decided to go for a 17-plex. So now we're going to look at some practical training. So whenever you are going to use a Bio-Plex assay, it's always good to plan, prepare, and perform. So review the protocols, set up instruments, gather supplies and samples. And it's always a good idea to have a look at what samples are recommended and which anticoagulant is recommended for use with the assays. And then you can run your assay. So when you're using the Bio-Plex 200, you have to allow 30 minutes to warm up. Those are for the lasers. Check the probe needles and clean if necessary. So if the bead rates during calibration should be between 200 and 400 beads per second with a clean probe. And then we have general maintenance of the instrument. So daily you would start up, calibrate, wash between plates, and shut down. Weekly, you would need to sonicate the needle, unclog and check for leaks. And monthly, validate and clean the exterior surface. And then probably yearly, replace the sheath filter and intake valves. So calibration and validation kits supplied by LASEC. So the calibration kit you run once a day before running an assay. It looks for temperature changes and use of other software for instrument control. Then you have the MCB plate, clearly labeled reagent locations, calibration on the left-hand side and validation on the right-hand side, routine washes, and probe height adjustments. So the validation kit you run once a month when calibration fails, whenever you want to confirm the instrument is functioning correctly. So here we have the Bio-Plex 200, the Bio-Plex 3D system. Then we have the Bio-Plex Pro wash station or the handheld washer. Then the ICA MTS 2 or 4 shaker set to 850 RPMs. And then you need a vortexer to vortex the calibration and validation kits. So the kits come with buffers and diluents. So remove your assay kit from the fridge at least 30 minutes before you wish to run it. Remove the buffer bottles from the packaging and allow them to stand on the bench, and place the standards secondary detection antibodies, SAP, phycoerythrin, and capture antibodies vials on ice. Then you would need water, sheath fluid, and reagents, extra black flat bottom plates, tube racks, titer tube micro test tubes, 1 mil. You would probably need aluminium foil just to cover the plate so that there is no light enters the plate. An ice bucket, ice, 15 mil tubes, 1.5 mil tubes at a time, and a permanent marker. Then you would use a multi-channel pipette for plate loading, single channel pipettes for sample and reagent addition, pipette tips, and a 50 millimeter reservoir. And then proper pipetting technique is important. So review the instruction manual before running the assay for the first time. Ideally, you should review before collecting samples. Keep the quick guide at hand. This is just a copy of the quick guide on the right-hand side here. And review instrument operation if you are new to Bio-Plex. So the main source of variability is the operator, inappropriate sample collection and storage techniques, resuspension of standards and cytokine assays, temperature variability of buffers and reagents, not vortexing your tube of beads sufficiently prior to use, poor pipetting techniques, inconsistent washing and or plate filtration, but we don't use the plate filtration anymore because we're using magnetic beads, and poor maintenance or upkeep of your Bio-Plex system. So good practice and troubleshooting. So follow good laboratory practices when running Bio-Plex assays. Always remember that reproducible data will require consistent protocols. Changing incubation times, number of wash steps, etc. will change the results. Immunoassays must be allowed to reach equilibrium during the association phase to give reproducible data. And if they are incubated for too long, the antibody-antigen complex may start to dissociate. For our assay wash steps, 300 microliter washes are not the same as one 300 microliter wash. So serum is preferred for cytokine as it generally yields better cytokine levels. If plasma is used, then heparin should be avoided as this interferes with the antibody-antigen interaction. ED3A and citrate are better. When whole blood is collected, it should be kept at 4 degrees C until processed to avoid hemolysis, which is the rupturing of red blood cells. Don't agitate whole blood, as this will increase the rate of hemolysis. So here you can see good serum on the left and bad on the right. For serum collection, whole blood should be allowed to clot for 30 minutes, maximum of 2 hours at room temperature before centrifugation to separate the serum from the red blood cells. And after centrifugation, transfer, mix and aliquot the serum into 50 to 100 microliter aliquots and freeze these at minus 70 degrees C. Avoid multiple freeze-thaw cycles as cytokines degrade significantly after 2 or 3 freeze-thaws. So centrifugation, centrifuge at 1000 times G for 15 minutes at 4 degrees C and transfer to a clean tube. Repeat centrifugation at 10,000 times G for 10 minutes at 4 degrees C, especially when using serum and plasma. This ensures complete removal of platelets and precipitates. For storage, avoid repeated freeze-thaw cycles, aliquot samples, and keep at minus 80 degrees. So our protocol advises that you resuspend the lyophilized vial in standard buffer or suitable alternative, vortex gently, and then incubate on ice for 30 minutes. We have observed in-house that our standard proteins do not resuspend back into solution at the same rate. And so it is necessary to leave them for 30 minutes to ensure they are fully resuspended. Leaving them for 2 hours is not recommended either. We do not recommend that you freeze any unused standard solution for future use because it is possible there may be some unknown level of degradation of the cytokines during storage and freeze thawing. Run all samples and standards in duplicate. So here we see when we resuspended for 15 minutes on ice, interferon gamma standard curve showed a lower limit of quantitation of 9.15 picograms per mol and an upper limit of quantitation of 41,311 picograms per mol. When resuspended on ice for 30 minutes, the lower limit of quantitation was 2.75 picograms per mol, and the upper limit of quantitation was 41.205 picograms per mol. So this change in incubation time led to a 3 times reduction in the lower limit of quantitation for this target analyte. So the association and dissociation rates for antibody-antigen complex are going to be influenced by the experimental conditions of the assay, such as temperature and buffers. If the assay buffer is still cold, then the association rate constant will be reduced. This means that the complex formation may not reach equilibrium during the 30-minute incubation step, so the data will not be reproducible between assays. It is best to always remove the assay kit from the fridge at least 30 minutes before you wish to run it. Remove the buffer bottles from the packaging and allow them to stand on the bench so that they can warm up. And the secondary detection antibody, SA phycoerythrin and capture antibody beads are kept on ice. Whilst you are resuspending the standards on ice, you can prepare the tubes you will use to generate the standard curve by adding specified volume of standard diluent to each tube. When you then add the resuspended standard to the first vial, you create an 8-point serial dilution, and this will warm the standards to room temperature quickly. Remove the beads from ice and vortex for 30 seconds prior to adding the required volume to the tube containing assay buffer. Protect this from light until it is needed to avoid photo bleaching of the bead. And it is best to use a P200 pipette to transfer beads from their tube and not a P1000, because the larger tip of a P1000 can cause a seal around the neck of the tube and lead to inaccurate volume being removed. So use calibrated pipettes. Pre-wet the pipette tip before use. Work at temperature equilibrium. Examine the tip before dispensing sample. Pause after aspiration. Hold the pipette straight, not at an angle. And minimize handling of the pipette tip to avoid contamination from your hands or gloves. And use the correct pipette tip for the brand and type of pipette you are using. And use consistent plunger, pressure, and speed. And avoid touching the bottom of the filter plate. If you're using the 96-volt filter plate from our touching the filter membrane with the pipette tip, this could leave a hole which will result in significant bead loss. So prerequisites for cytokine measurements in clinical trials with multiplex assays. So generally most cytokines will start to go down, but there are a couple, interleukin-4, interleukin-12, TNF-alpha, and interleukin-18, which tend to go up. And I'm not sure why that is. Optimal shaking conditions. So when shaking the beads, it is recommended for the recommended setting of 850 plus minus 50 RPMs. Must use an orbital shaker, and the shaking the plate at room temperature. So this is where you can get an ICA 2 or 4 plate shaker from LASEC. So long-term stability, we've seen that most of the assays with cytokines go down over time, but we have seen earlier that some of the assays can actually go up. Sorry, I'm just moving my... So can I increase the sample incubation time from 30 minutes to 2 hours? Change the sample dilution. So for serum and plasma samples, it's normally a 1-in-4 dilution. Running my assay at high PMT and adding a 0 point to my standard curve. If anyone wants to come off mute, the correct answer to all these questions is no. So here you can see 30 minutes at the top, 2 hours at the bottom. And here you can see 0 to 50 on the concentration in range. And by increasing the detection hour to 2 hours, you have limited the concentration range from 0 to 15. And here, 30 minutes to 2 hours, human interleukin 16, human interleukin 16. So you can see there's no real difference between incubating these samples for longer than 30 minutes. So if the protocol says 30 minutes, then just incubate it for 30 minutes, and you can't increase the sensitivity by incubating for longer, for like 2 hours. Here we have serum and plasma. So it's normally a 1-in-4 dilution. And you can see adequate levels in the blue of being red. But if you do a 1 in 16 dilution, then you can see that the analytes have actually gone up from the blue graph. So if it says a 1 in 4 dilution, then please use a 1 in 4 dilution. So some customers like to use a high PMT, but you can see that the upper limit of quantitation is 3,505, and the lower limit is 0.196. So if you're using high PMT, it gives increased assay sensitivity and an improved lower limit of quantitation compared to running at high PMT. So most of our Bio-Rad's assays are running at low PMT. So some suppliers say that you can increase the accuracy of the standard curve by drawing a position to naught, but that's mathematically not correct. So it's better to have the proper assays is to use sample 1, 2, 3, 4, 5, 6, 7, and 8, your standards, and not to do a standard, produce it to naught, because that's not mathematically correct. And then if you're looking at the standard diluents, the upper limit of quantitation is 4,174. The lower limit of quantitation is 0.016. So that's in standard diluent. If you're using PBS, you can see that you've now reduced your upper limit of quantitation, and your lower limit of quantitation is increased. And if you're using sample diluent, you can see that your upper limit of quantitation has dropped even further, and your lower limit of quantitation has dropped as well. So I just wanted to say thank you to everyone for joining, and that is the end of today's presentation.